Hello everyone, today we'll be talking about chemoreception, the senses of smell and taste. First, we'll do an introduction of chemoreception. Chemoreception is the process of responding to chemical stimuli through the senses of taste and smell. Chemoreception is critically important in sensory perception for several reasons. It's used in the detection of food and toxins, social communication in certain organisms, and via pheromones. Awareness of surroundings, such as predators or marking territory boundaries. It's also involved in memory or learning, and health and pathophysiology will also be discussed later in the presentation. We'll first speak about olfaction, which is a sense of smell. The anatomy of the olfactory system includes the olfactory epithelium, which is located in the upper part of the nasal cavity. The olfactory epithelium is a specialized tissue that contains olfactory sensory neurons. The olfactory system also has the olfactory receptors, which are proteins located on the cilia of olfactory sensory neurons within the epithelium. Each olfactory receptor neuron typically expresses a single type of olfactory receptor protein, but different neurons can express different receptor types. These receptors are highly specific and can detect specific odor molecules. The olfactory system also includes the olfactory bulb, which is a neural structure located at the base of the brain, just above the nasal cavity. It receives input directly from the olfactory sensory neurons through bundles of axons called olfactory nerves. Within the bulb, information from different olfactory responses is processed and integrated. It then projects this processed information to a higher region, including the olfactory cortex, where further processing and interpretation of smell occurs. Mechanism of olfactory transduction includes the binding of odorants to olfactory receptors, activation of G protein-coupled receptors, and generation of action potentials in the olfactory sensory neurons. Specifically, the pattern of receptor activation is the odor binds to olfactory receptors located in the olfactory epithelium and the mucous membranes in the nasal passages, which activates the olfactory G protein, which then activates adenylate cyclase. The adenylate cyclase converts ATP to cAMP. Then, the cyclic nucleotide-gated channels open, causing an influx of cations, which causes action potential to occur, which signals a neuron to the brain. Activated olfactory receptors form specific receptor codes that are transmitted to the olfactory bulb. The olfactory bulb receives these signals and expresses receptors and then transmits the signal patterns to dendrites of the mitral cells, which take the signal to the olfactory cortex, which allows the brain to process the sense of smell. The olfactory system has direct connections with the limbic system, which includes structures like the amygdala, hippocampus, and hypothalamus. The amygdala in particular is involved in processing emotions and emotional memories, while the hippocampus is crucial for forming new memories. In this process, olfactory signals can evoke strong emotional responses and trigger vivid memories. It can also evoke memories of specific events, places, or people, which is also called associative memory. The sense of smell is often more strongly than any other sensory cue in invoking memories. And we will talk about gustation, the sense of taste. The anatomy of the gustatory system includes taste buds and papillae, which are further divided into the fusiform, circumvallate, and foliate. There are also gustatory receptor cells, and there are also involvement of cranial nerves, such as the facial nerve, glossopharyngeal nerve. Taste transduction is an interaction of chemicals with the receptor cells. The signal transduction pathway is involved with ion channels and similarly the G protein, the GPCRS, which also causes a generation of graded potentials and action potentials. Taste perception is a little different. Taste is differentiated by taste quality such as sweet, salty, sour, bitter, and umami. The neural processing in the gustatory cortex is a little different than how smell is processed in the olfactory cortex. There's perceptual aspects that are a little bit different. Role of taste is involved in food selection and preference. There are cultural and individual variabilities in taste perception, such as different cultures have distinct culinary traditions and preferences shaped by cultural practices, availability of food, and social norms. For example, Indian cultures use a variety of spices to create their foods. There are individuals also who are particularly sensitive to certain tastes, while others may have reduced sensitivity, affecting their food preferences as well. Olfaction and gustation have some similarities and differences. We see that receptor types and mechanisms of transduction vary with olfaction and gustation in certain ways, and there's differences in their neural pathways and cortical processing. In terms of similarities, detection of chemical compounds are similar in both olfaction and gustation. Both olfaction and gustation detect chemical molecules, odorants and tastants respectively, and they're dissolved in surrounding medium, whether it be air for olfaction, saliva for gustation. They rely on specialized receptor cells located in their sensory organs to detect specific chemical stimuli, which is also a common similarity between the two. In terms of signal transduction, both olfactory and gustatory receptor cells use specific receptor proteins to detect and bind to their respective chemical stimuli. They both involve GPCRS or G protein coupled receptors in their signal transduction pathways through different types and specificities. Then both oral and gustatory information is transmitted via cranial nerves to the brainstem for an initial processing of whether it be smell or taste. There are several differences between the two. In regards to receptor types and mechanisms of transduction, olfaction receptors are GPCRs that detect volatile molecules like odorants based on their chemical structure. The mechanism is a little different. They are receptor proteins leading to the activation of G protein signaling pathways, which is the adenylate cyclase and the CMP pathway. In gustation, we see taste receptors are diverse and include GPCRS sensitive to specific taste qualities. So there is a difference using varying sweet, salty, sour, bitter, and umami. The tastants bind to receptors on receptor cells within the taste buds, triggering the signal pathways such as activation of ion channels, like the sodium channels for salty taste, potassium channels for bitter and sour taste. And their neural pathways and cortical processing are different. Olfactory information is transmitted via the olfactory nerve, which is cranial nerve 1, to the olfactory bulb, then to the olfactory cortex. In gustation, the taste information is transmitted via the facial nerve for anterior, the cranial nerve for anterior tongue, and the glossopharyngeal nerve for the posterior tongue and throat, to the brainstem in the thalamus and the gustatory cortex. In terms of disorders and impairments, we see anosmia, which is the loss of smell, and ageusia, which is the loss of taste. Anosmia can result from various causes: blockage, like obstruction from the nasal passages due to polyps, sinusitis, or physical injury. There's also damage to the olfactory receptor neurons, which could be due to viral infections like the common cold or COVID-19, or toxic exposures can damage or destroy olfactory receptor cells, impairing signal transmission. There are also neurological disorders such as Parkinson's disease, Alzheimer's disease, or even trauma can affect the olfactory nerve fibers that can affect the central processing. In terms of agousia, which is the loss of taste, it can be caused by several factors also, such as damage to the receptor cells. Viral infections, medications, head trauma, or toxic exposure can damage or destroy these taste receptor cells. There's also neurological conditions involved, but the conditions would be more like Bell's palsy, MS, or stroke that can affect the facial nerves or the glossopharyngeal nerve that transmit the taste signals. In terms of agousia, dental issues, oral infections, or dental procedures can temporarily affect taste perception. If there are any deficiencies in zinc or vitamin B12, that can also alter the taste perception. In conclusion, there's key points of chemoreception. Understanding these key points helps elucidate how chemoreception in smell and taste functions at the physiological level, contributing to our sensory experiences and our interactions with the environment. The senses of smell and taste are not only essential for survival and health, but they also nurture daily experiences. How we smell things, how we eat, how we have preferences for food. It's how we interact with the world around us. These roles extend beyond just mere sensory detection, and they encompass emotional, social, and cultural dimensions of our life as well. Thank you so much for listening. Have a great day.